Oh, wow.
And it's got all the anemones in the lee right there.
Yeah. Are those puffball anemones? Is that the common name, or is that just what I think of them?
Pom-pom, I think.
Pom-pom, there we go.
Yeah. Lipanema. Clearly loving this habitat as well. It's definitely Cnidarian's rule here. There's a few sponges, but fairly small.
The center screen just above the lasers is called a mushroom coral, or heteropolypus. We do have some inverts. We have decapods, we have that crab to the left, the rockfish.
Small snailfish.
Oh, and yeah, a snailfish on the rock to the left that we just passed. For anyone just joining us, we are doing a transect near St. Lucia Bank, just northwest of Point Conception, which is a very well-known biogeographic break.
This area would be included in the proposed Chumash National Marine Sanctuary. So we're doing some surveys to find out which corals and sponges are associated with different habitats down here. We know it's an area where there's diverse and abundant marine mammals and seabirds.
Yes,
Come
very
to feed
productive
around the bank.
surface.
Mm-hmm.
And so we were definitely curious what we would see at the bottom. And it definitely depends on which part of the bank you're on. We've seen a pretty diverse array of C4 habitats and very small, not so many corals and little sponges, to now what you're seeing on your screen, which are very relatively larger corals, big boulder habitats, high current, lots of food for the filter feeders. This is so exciting. It's like Christmas. Flatfish.
Deep sea sole.
Awesome. I'm terrible at my fish IDs.
That's the one with the really distinctive grey pattern.
Yeah.
Almost checkerboardy, but Erica always says she wants wallpaper in deep sea sole.
Hard in there. Can I take it out of auto XY and then get it up?
I see a lot of.
Oh, I turned the
Yeah, you gotta get going again though.
Yeah, yeah.
Looks pretty steep coming up
just
here.
Continues on like this. Quite a ways. Very neat
different
rocky terrain. Lots of slabs and boulders, blocks.
given how strong the current is here, if we were to go back to that other spot, how hard would it be to do something like a photomosaic, a small one?
We're about 60 meters off from Coral Garden right now. Oh, but I guess you're saying at the end of the transect, so I'll let
Or just
defer to
in
the
theory, like if we weren't to go back there, but we did see something kind of similar where it's not super steep, like given how strong the current is here,
Well,
I wouldn't want to ask.
we're about to see a lot of
something a little-
corals when we get
Yeah,
up
look
to
at
the
that
top here
Argus
too.
view.
But you need to hurry up.
Yeah,
It's
yeah.
almost like a drop cam. If you could put lasers on it, you could use that for photo
Oh, that's
quadrats.
great. Yeah.
Wow.
Yeah, so anyone that's joining us who's just watching HertCam, it might also be if you open up another window or switch over quickly to the Argus Cam, which is channel 2, or the Quad, which is channel 4, you can see a lot of the corals from a higher perspective, so you have a broader context of how many corals we're looking at, which is stunning.
Yeah, it's definitely steep. This looks like, from our slope map, we're getting near the top of the slope. It's a little hard to tell. The bathy suggests that it's almost like a plateau up here.
I wonder if there might be.
There's actually a nudibranch here.
Oh,
This is-
yeah,
Yeah.
is that Tritonia?
Pull up.
Just to upper left
What's
of the
happening?
lasers.
I'm full throttle up right now.
I think just the current's coming down the wall. I think
Yeah.
must be.
Yeah. Should I slow the ship down?
Yeah.
Yeah.
Help.
We're struggling here. I don't think it's too much higher, but
We're going to slow to 0.2. Let me know if you want to drop down lower. Yeah, we should.
yeah, he's gonna just come out stop because he's not making headway here. We're gonna fall off and get
Bridge,
yanked.
nav. Hold position.
And this is full throttle, right? I guess there's still a little bit of Z bias.
Well, if you got 100% thrust, then
Yeah.
you
It's
got
100%
100% thrust.
Is this something where we would need to zigzag to get upslope?
Sorry, can you repeat that?
Just in terms of strategizing to get upslope, is this something where we would like do cutbacks, zigzag our way up?
I think we're still making it upslope now, just slower than the ship was moving forward, so.
Got it. Okay.
Definitely a lot of skate A cases. Wow. Just 1, 2, 3, 4, 5, 6, just in this frame. 7.
And you can see on that coral to the right, there's no polyps covering its base, and you can kind of see that segmented
Oh,
portion of its
the size of that paragorgia
skeleton.
base, that's nuts. It's 15
Oh, wow.
centimeters.
This is a much steeper slope than anticipated.
I just can't believe this current.
Yeah.
Well, that's why they're here.
Yeah.
You
How
can
far
see
is
it's
that
all
trend?
bent over down the slope, right?
Yeah. I mean, this is full throttle.
We're about halfway through the transect, and we're about 20, 30 meters from what looks like the end of the slope. We should be peaking.
Yeah, that sounds great. The distance is more important than the time it takes to get to 200 meters, so this is just fine.
We're at the last contour before it starts leveling out, but maybe that's why we're getting even more current. Wow. Yeah, you're just
Oh, nice.
not moving.
Yeah,
Incredible.
just for reference for everyone at home, this is full throttle up right now, and we're barely climbing, so.
Wow. See a large heterocone sponge there, that orange pitted sponge to the left.
It's back again.
Yeah. Is he going in there?
He's just, I don't know what he's doing. But he's squeezing through the gap, you know, trying to put something in the overhead.
Yeah.
Everyone in the science chat is rooting for Herc. You can do it, Herc.
A little ROV that could.
So how fast do we estimate these currents to be traveling then?
No idea. We don't have any sort
Okay.
of way to gauge it. The
Got
only
it,
way to
okay.
gauge it's to just quit thrusting and go with it, you know? We don't
Oh.
want to do
See
that.
how fast you're traveling? No free falling today.
How many thrusters does Herc have?
Not enough for right now.
So it has 6 thrusters, but really 2 of them are pointed up, so. But they're big thrusters, so.
We go down better than we go up because of the box on the starboard side. The thruster blows against it, so it kind of decreases our efficiency going up.
Mm.
I really don't like that he's right there. That's
Do you want to?
You want me to step
You
back?
can tell George. No, it's.
Oh, sorry, in the winch. I can ask the bridge.
well, yeah. I mean, yeah.
I mean, he knows it's running.
Sergey's been here since 2010. He's pretty sharp. Bob, you are pointed all the way down on Argus, right? That's a hell of a view.
What?
Argus, pointed all the way down.
It's all the way down.
Thank you.
That's all you get.
It's kind of like my zooms, huh?
Yep.
And how much longer do we have roughly within this dive before we need to pull to be back on deck?
We have,
We're estimating in the science chat.
sure, we've got, let's see, give me a second to calculate that. Well, it'll take us about an hour to hour and 10 to get to the surface. Right now it's 9:30. So we'll need to leave by 10:45-ish.
Okay. Okay, excellent.
And
Thanks,
from
Di.
3F
10:45.
Yeah, and then from 3F to 3G, that whole span would have taken about 45 minutes if we had just been moving steadily at 3 knots. So I think we're actually making pretty good time if we have an hour left on the seafloor.
Okay.
Bob, I think we're looking good in the winch room, yeah?
Yeah, well, hopefully he's done doing whatever he was doing down there.
Yeah.
So for anyone watching, we are currently transecting up a steep slope on Santa Lucia Blanc at about 900 meters, and we've encountered some pretty strong currents coming down this wall. So just making our way up gently through this coral garden of bamboo corals. I think there's at least 2 different types of bamboos and some bubblegum corals as well. A few glass sponges in the mix. This looks like some big sponge here.
Yeah, Steve, what are the different parts that make up the bamboo skeleton that actually looks like vertebra? What's the different components there?
Yeah, most of the bamboos are made of, the skeleton at least is 2 parts. It's got a proteinaceous node, which is the dark band that goes all around. It provides some semblance of flexibility to the colony, and then there's the calcitic internode, the white kind of bony fragments.
Our clip came off.
Little branching asbestos pluma.
Dang
We
it.
were on the lookout for those.
Whatever.
And what is the internode made of, Steve? Is that calcium or is it also flexible?
Yeah, the bamboo corals are, they're high magnesium calcite skeletons mostly.
Okay.
Most octocorals are calcitic, but that's not always true. There are some octocorals that do produce arachnitic elements of their skeleton.
And Lisa Winnick, who is the author of this dive plan, is saying no need to make it to 3G. It's like kind of how both of our dives have been going is the waypoints were suggestions and we're mostly looking for interesting habitat so we can decide how to proceed maybe once we get to the terrace or the precipice of the slope and give it a look around.
Roger. Definitely gonna take us a little time to get up there.
Yeah, I imagine we have some time to think about it.
Sure do. Lots of sponges.
Looks like maybe we're getting to the top here.
Yeah.
Come on, Herc.
Do you think the current will be less extreme once we get to the terrace? Or do we think it will be the same?
One way to find out.
Yeah, that is very true. A lot of NA sponges here.
I imagine it's flowing over the top and then down. So it's probably going to be, but it's going to change from downslope to in our face.
So interested why skates would be laying their eggs in such a high current area. I think they're able to attach a little bit there, but very interesting.
The water is also very clear compared.
Yeah, now it is. The base of it was actually pretty turbid. I'd be very curious to see what the predictive habitat models say about this area.
That's an excellent point. We're actually at 900 meters, so that should be within the range of those models. They originally had a 1,200 meter depth limit for a number of different reasons. Just the observations were pretty sparse.
So we still got transit left?
Oh yeah, we've got.
You could probably give her the beans again because we're on top.
Yeah, we've got 80 meters left of the transect.
Yeah.
Yeah, I think.
Are we ready for a ship move?
Yeah.
Bridge nav. (100 meters, bearing 0). What speed do we want to go? We want to.
You could. I think you could go back as long as we don't see anything coming up slope-wise.
Okay.
I would think.
Bridge nav. We can bump back up to 0.3 knots.
Still a pretty good current, but in our face instead of blowing down.
Yeah.
Got more thrust going ahead than up.
Yep. Yeah. So this should be a little bit of a plateau here. Leveling out.
Kind of out in front now. Getting kind of.
Yeah, yeah.
Large bamboo skeleton.
Yeah, I think you have to go slow because you're way out in front.
Yeah.
Do we have any guess on what the white, gray-ish encrusting organism is on a lot of the rock faces here?
I was just wondering that myself because they're only facing, they're not into the current, they're protected. So I'm wondering, maybe a colonial tunicate, because they could potentially be so soft they wouldn't be able to survive the current. I'm sure.
Yeah, it'd be worth a zoom when we're off transect if we see it. Could be encrusting sponge, encrusting tunicate.
And I definitely see upper left that candelabra morphology that you were talking about, Steve.
Yeah.
A lot more sponges up here too, Abby.
Candelabra
Lineas
Isabella.
everywhere.
Yes, yep. Very glad we got a nice sample earlier. Maybe a yellow Eucalyptus coming up on the right. Big patch of barrier. More of these big slabs, which Guy Cochran from USGS was explaining, pieces of broken slab that have come off, fractured off the main bedrock of the bank and are kind of falling down the slope.
I was just about to ask Guy that, Abby. Excellent. Piece of information.
It's always good to have the geologist
Big
perspective.
numbers.
Is this where you use the claw to grab onto the corals and use them as handles to pull Herc up?
Actually, now we're able to make some progress, but Herc is a little bit out in front of Argus now, so just kind of
Okay.
going slow and letting Argus catch up a little bit.
Stealing your camera.
Yeah, go for it.
If anyone's on the quad view or the high pack channel 3, you can see the progress we're making up this slope where there are depth contours in white and then the slope is illustrated by these various colors, and that yellow red was the steepest slope, and now you can see we're in this like purple area where the slope is much less extreme.
You can also see the little zigzag where we started hitting current.
Oh yeah.
Yeah.
We're about halfway along our transect.
Not necessarily the cleanest transect, but definitely an interesting one.
Yeah.
Oh yeah.
And we've got about 40 meters left to go on this transect.
Thanks, Sam. Yeah, so this area of the California Current off of the west coast is known to be a particularly productive one. And so it does not surprise me that we're seeing a lot of this particulate organic matter flowing through the water. You can see the current as it rushes by, which means there's a lot of food for filter feeders like these corals and these sponges that we're seeing. Especially the deep sea corals and sponges at large, they tend to provide habitat for other species that humans are quite interested in, such as commercial fisheries. We have rockfish. There's a lot of flatfish in these areas, as well as invertebrates. There's a lot of crab, shrimp, maybe not in this particular location that you're looking at, but in general, deep sea corals and sponges are considered essential fish habitat. And so understanding where they occur and why is really helpful for not only protecting and conserving these long-lived, really slow-growing invertebrates, but also fishery species that rely on them to grow and reproduce for food, shelter. And it may not be that you're seeing these species in a life stage or at an age where they're being captured, but if they spend any of their lifetimes within these to grow or reproduce, they're going to be important even if they're caught elsewhere.
Yep, it's a question we're still unraveling. So what we think at the moment is that some of these associations are pretty species specific. We know, for instance, that some of the glass sponges are important for juvenile rockfish. But yeah, those relationships sort of vary in different areas.
Yeah, they think that they're more tightly linked the further north you go. But this will be really helpful information in terms of teasing those patterns and relationships out.
Mm-hmm. Oh, Lizzie, Guy has come in with some more information on the geology there for you.
Oh, I see. So the tectonics of this area, or the large plates underneath this habitat, have east-west compression and extension, and then northeast-southwest strike slip, so there isn't any unfractured bedrock. I think I have a general understanding of what that meant. It sounds like there's a lot of movement. Yeah, movement in multiple different directions, which is why some of those large pieces were broken up, because a lot of movement in the plate tectonics here.
So, and just for reference here, we seem to found a little dip, so we're going back downhill, which is why it seems like we're further from the surface, but it looks like we're going to be going back.
And the transect's actually complete, so we can stop here.
Awesome.
Alrighty.
Thank you.
Transect 8 is complete. What's next, science? As Paul mentioned, we're starting to head back down, still towards 5G, but downslope. So
There's going to be a little knoll right ahead too if you look in sonar.
Yeah, you're right. Let me see what Bathy says. If there's any there.
So we have a little bit less than an hour left.
That's correct. So we definitely have time to get to 3G if that's still of interest. Finish that set of waypoints. And
so yeah. Mm-hmm.
If you're looking at HYPAC right now, you can kind of see that little knoll that Paul's talking about on the starboard side of the ship in a perk.
Yeah, so if we were to go like to the left, I'm looking at HYPAC now, it looks like we could have a little bit more of a slope. And we wouldn't be directly going to 3G, but we could follow where the slope is steeper up to the upper left. Or, so I think we could keep going up. I think we kind of have two options. We can keep going up, but follow like maybe the steeper slope or where the contours lines are the closest. Or we could go, if it's a safe maneuver to do so, to kind of head back down the slope and come up parallel in another area and do another transect, or do some, I don't know, if the current kind of seems too high to do any collecting to me. Do you have the same sense?
I lost part of that. What was that?
Yeah, I didn't follow what you were saying there.
Okay, sorry about that. So we could either go towards 3G, but follow the steepest slopes, so not directly go towards 3G, the last waypoint. Or I also saw in the science chat some suggestions to go back to where we dropped the waypoint when we first came across the coral garden to do more exploration there.
Are we going to be able to get back there reasonably?
Well,
yeah, and I'm also wondering.
I mean, it'll take you right back there.
Yeah.
Might be
safely. Keyword. How do we do that
just
safely?
let go and we take you right back there.
Yeah.
So Lisa Winnick is saying we can go left around the knoll to follow the steep slope up to the terrace where 3G is. So we wouldn't be going directly to 3G, but we would almost be following the contour that we're in now.
Roger.
Like northwest, and then go up the slope where it's steeper.
Roger, sounds good. So the direction the ship's pointing is where we'll head. Perfect.
Yeah, we don't want to push Herc, and we don't want to get ourselves in any tricky situations.
Agreed.
What they mean?
Bridge nav. 30 meters, 315.
Is it possible to zoom out a little bit on HYPAC?
Yes.
Just a little bit.
What was the bearing?
315.
315.
Oh yeah, Kurt Whitmire in the science chat was just wondering if 3G is at the very top of this feature. But to me, it kind of looks like it might be just the first.
The start.
Terrace at the very top. It may keep going. I can tell.
But pilots, your heading might be more like 330 to look at the slope as we get near it.
You're yanking on me.
Oh. Yeah, I mean, I need to come back.
Yeah, you gotta kind of zip over there.
Yeah, that was like full lateral over, and I just still kind of got blown off screen.
Lizzie, just to confirm, I am replaying what we just said in my head. We wanted to go kind of left up here, right, to the northwest?
Yeah,
Okay.
yeah, head northwest until we can go more northeast up, pretty much up, straight up slope.
Roger, thanks.
Yeah, and Kurt is echoing. Once you get to a point where the contours indicate a steeper slope, head straight up towards the top.
Great.
Do we need a DVL reset there?
Yeah, we probably do. Want to wait till we stop or do it now? We're about here.
Yeah, you're getting yanked. You got to get back under.
Which way do I need to go, Bob?
Well, you need a lateral. See, just lateral over here. Like,
Yeah.
We do a reset now?
yeah.
There we go.
Back into to cobble with some small sponges. By the sound of it, still strong currents.
There you go.
Yeah, yeah. Now I'm going.
Some of these unbranched crinoid corals, possibly Narella.
That might be a little snailfish or no, Bob?
Yeah, I think so. I think a little snail.
Okay. Oh yeah.
It was
yeah,
the
that's right,
rosy snailfish, smaller than the blacktails.
What time is it now?
Yeah, on deck at noon. Right now it's 10.
Okay.
So we're gonna off bottom no later than 10:40.
So are we going northwest and then northeast to try to dodge the current?
Yeah, we're trying to kind of move up the slope this way versus going straight into it. Do you think that's working for you?
Yeah, but we could if we're in a time rush, we could just go straight for it as well.
Yeah, I don't think we need to try and.
I think the current's going to be here either way.
Yeah, I agree.
So we could just go.
What about?
Oh, yeah, go ahead.
Sorry, I was just wondering about in terms of interesting habitat. I think we were going to try to target some of the steeper slope to see if we might find more corals.
Oh yeah, yeah. As long
because
yeah.
3G in particular is not. It was like a guiding point versus like something we needed to do there. So.
All right, as long as there was a science objective, I just wanted to make sure it wasn't just for the current that we were going over there. So that sounds good.
Yeah, awesome. Thanks for communicating that.
I just got here. Are we on transect?
We're not.
Thanks.
I'm looking out for any of those branched carnivorous sponges.
Oh yeah.
I was seeing quite a few on the previous watch, and I think I saw one when we were coming through that very high current area. But definitely not so common here. A mushroom coral. Little thorny head. Some of that unbranched Primnoa.
Yeah, we're seeing a lot more sponges now.
Mm-hmm. I was trying to work out if we're seeing more sponges or if it's just that the sponges were there, but we were quite distracted by the corals. We were surrounded by those huge bamboos.
I feel like there were more on the upslope. We saw more sponges than we did when we initially saw the coral garden.
Yeah, definitely all those near. I mean, both corals and sponges like areas of high current because it brings them food, but they do feed on different
Again, way out in front
components
again.
in the water column.
Yeah.
Corals tending to trap larger particles than the sponges. They're filter feeding.
Yeah, and some of you might be more familiar with shallow water corals, which rely more on their symbiotic relationship with phytoplankton within their tissues that provide them with nutrients as they photosynthesize. But these corals are also known as cold water corals because they're so much deeper, they do not rely on a symbiotic relationship to survive. They rely solely on filter feeding. And so these high current environments, like Abby was mentioning, are really important habitat, or a really important aspect of their habitat. Oh, it looks like a hagfish at the bottom of the screen.
Yep, the deep water corals often have shallow water cousins. Then you get some bamboo corals on coral reefs that have the zooxanthellae, the little algal symbionts that provide them with food through photosynthesis. They look quite similar, actually.
Yes, but we don't have, oh gosh, what is it called? The massive corals, like the big ones that make mounds, those tend not to be found in the deep sea.
Oh yeah, like kind of the ones that look like brains,
Mm-hmm.
big massive corals.
Yeah. And if anyone listening is interested, we're having Nautilus and Ocean Exploration Trust are having a Next on Nautilus live event today at 1 o'clock. So I encourage you all to tune into that.
And you can find links to that event on the Nautilus Live website or on Facebook or YouTube. And
Sam will be there.
I'll be there. Lizzie will be there. Lisa, Julie, and then Violet Sagewalker from the Northern Chumash Tribal Council.
And what time again?
That'll be 1 p.m. Pacific today. You're also on the invite, Al, but you won't be on camera.
Oh,
I know.
but we'll be here at 12:30 to get testing, right?
Sure will.
Another yellow Storocaliptus.
Are we seeing some asbestoplumas, like the unbranching forms, or are these corals still?
I think there's definitely some of the asbestopluma in the mix, like that one there next to the Thinea, I think. There's asbestopluma.
Okay.
I'm looking at these, the white sponge just going off the screen at the moment, trying to work out if this is what I sampled on the first shift, or we sampled. How does the current feel here? Would this be, would it be possible to take a sample in these conditions, or is it
Yeah,
too intense? Okay.
I think so.
The struggle was when the current was coming down the slope. You know, you're trying to drive up into it. You don't have as much thrust. You have more surface area. You're trying to move through the water, and you don't have as much thrust going up. So we're driving ahead up a slight slope like this. It's okay.
Are you looking to take a sample
Got
here, or?
No. I was just asking for if we see something. It's just good to know.
Yeah, I think if you see anything, just give us a shout.
Yeah.
We have several dead sponges here too. Almost look like rock, but they're much more porous.
It's like there are some skate eggs that have been laid on top of that glass sponge straight ahead.
Yeah.
I've never seen this many before.
Stuck all over it.
Wow. Any elasmobranch people on the side chat who can?
Let's get a zoom on these eggs for Sam.
Not for me. So interesting.
All right, let's come wide.
Interesting crabs around here too, and a shrimp.
Well, we've been seeing a lot of the rough-tail skate. I'm not sure what their egg cases look like.
Another skate egg on the ground there. A tiny rockfish, that little red fish in the center right screen.
Oh, yeah.
Lizzie, I think your mic is hiding again.
seen a few rockfish, not a ton, but we've seen several. There's 2 more there on the left. Another important fisheries off the west coast. And we have recreational and commercial for rockfish. It looks like Garcinia right in front of, just below the lasers. There's a fox coral that goes in the deep sea.
All right, Al, let's get a quick zoom. All right, let's come wide.
Soft corals definitely seem to be much more abundant than the stony corals here. Steve, is it cup corals that are most common of the stony corals we're likely to see at these locations?
Yeah, definitely. I haven't seen very many stony corals though. Really, at any site. Strangely enough, we actually did sample one yesterday at Davidson at 3,200 meters, cup coral, which was extremely brittle.
Oh,
It's
nice.
alive, so that should be useful. Yeah, you're not going to see corals like Desmophyllum until you get a little bit shallower, and even Lophelia, you have to get even further shallower, probably only a few hundred meters maximum to see those kinds of corals.
More skate eggs on a sponge. Is this an
Look
association?
at that. Yeah, I think it is. It's a new functional role of sponges in the deep sea.
And it's on the outside. They're not even inside the sponge.
Mm-mm.
I wonder if it's just getting them into the current.
Yeah, I wonder if more like water flow is good.
I feel like that's a Dr. Seuss book. Skate eggs on a sponge.
Super interesting. There's, yeah, bottom right corner, there were some more skate eggs on a sponge.
Well, I mean, they do get used by other species.
Yep.
I love the fact that some dolphins will stick sponges on the end of their noses. I think it helps protect them when they're foraging around for looking for food.
Interesting.
This could be rubbed eucalyptus. We think this shelf sponge is likely Poecilastra. More of these Thaenia sponges we got a nice sample of earlier.
Are these all glass sponges mostly?
So the Poecilastra is a demo sponge.
Okay.
I think the Thaenia as well. So the glass sponges are the sponges
Sarah.
that have these.
At what point are we going to switch to going northeast?
Science, do we want to continue along this contour or head northeast to 3G?
I don't think it matters. It looks like even if we went a little bit further, the slope would be roughly the same. So I think we're ready whenever you are.
Roger. Bridge, nav.